Going live. Okay, Heidi.
Welcome everyone. Welcome to the second week of our Mid-Atlantic Micro-Nano Alliance seminar. We're glad that you can join us again from YouTube. And Ryan will start us off this week again.
All right, so hi everyone. Today we have Lynette Naler, who's going to be talking to us about her work. So Lynette graduated from Johns Hopkins University with a degree in chemical and biomolecular engineering and computer science. After that, she spent 2 years performing immunology research at the NIH in Dr. Jyoti Sen's lab. Currently, Lynette is in the 4th year of her PhD at Virginia Tech. She is in Dr. Chung Liu's group, where she utilizes bioinformatics and microfluidics to analyze the epigenome. She has been published both for benchwork and computational research.
All right, thank you very much, Dr. So Cool, for that introduction. As before, my name is Lynette Naler, and today I will be talking about how we've profiled cell type specific epigenomic variations in pre-cancer human breast tissue through the use of low input microfluidics. Now, I know that sounds like a lot. So I'm going to start with what is epigenetics. So you have different organs in your body. You have your heart, your lungs, your liver, and they all have the same DNA. But what makes them different from one another? Why does a heart become a heart? And that is partly because of your epigenetics. Your epigenetics can affect gene expression without changing the DNA, which allows you to have the different parts of your body. Now, your epigenetics can also be affected by the environment. For example, when mice were exposed to vapors before being shocked, they understandably started becoming afraid every time they smelled these vapors. And this fear response was even passed down to their grandchildren who had never been exposed to such treatment before. Furthermore, epigenetics are reversible. So cancer often makes use of this fact by removing or adding modifications to make it more effective. However, we can actually use this as a source of treatment. We can stop these changes and help stop cancer in its tracks. So overall, the epigenome is the heritable set of reversible modifications that can affect gene expression without changing the DNA. So there are many types of epigenetic modifications, but for today's talk, I'm going to focus on histone modification. You have 2 meters of DNA in each of your cells. In order for the DNA to fit, it has to coil into chromatin. It's tightly wound. And part of this process, the DNA wraps around these histones, these group of histones, and these histones have tails, and these tails can be modified. These modifications can affect the way genes are expressed, either activating by increasing transcription or repressing transcription, so these genes aren't as active. So the question then becomes, how do we determine which histones are modified? And we use chromatin immunoprecipitation. It is a method of using antibodies to isolate DNA that is attached to a protein of interest. We first take our cells and we break them apart to release all the chromatin within. Next, we fragment the chromatin into smaller sizes. Then we expose the chromatin to antibody-coated beads. These beads bind to our histone modification that we're interested in, and we can then separate these sections out. Then we remove the DNA from the histones and we can sequence it. Traditionally, this method requires 1 to 10 million cells per assay. So what does this data look like once we get it back? We get back these sequences of the 4 different bases, and we first have to align them to the genome. We look at a reference genome and we say, this sequence matches this area. If we see a lot of sequences in a given area, we can call peaks, which are shown in the purple images. These peaks are regions where we can say there is definitely histone modification occurring. And finally, depending on which modification we're looking at, can tell us a lot about the underlying biology. Also, where in the genome is it located? Is it near where genes are just transcribed, or is it far away? And we can determine also the importance based off of, are there more modifications in one sample versus the other? Or are these modifications near genes that are important for certain pathways? Well, like I said before, it requires a lot of cells. So how are we able to minimize that? Because there's a lot of biological questions we can't answer if we require 10 million cells for each test. And to do that, we use microfluidics. Now, microfluidics has multiple benefits. First, it has a very small length scale. This can actually be leveraged for more effective assays, such as in adsorption, which is critical for the chip process. Because of their size, they also have small reactor volumes. The reduction in volumes allows us to have less reagents, which means less cost, and also allows us to reduce the amount of sample input. Finally, the nature of microfluidics allow us to integrate multiple assays into one device. You hear this frequently as lab on a chip because we can do multiple tests, run a lab on one device. In our lab, we created the microfluidic oscillatory washing Seek device, the MOWChIP. On the left, you can see an overview of our device. It is a 2-layer PDMS device. The main layer is a single channel with lots of columns to prevent the device from collapsing. And we also have a valve that can close the outlet. We can control the valve with solenoid valves and compressed air, and we can flow in materials to the device with a syringe pump. As you can see on the right, these devices, when they're made, are kind of a tough rubbery material. And so we use a fresh device for each assay, which prevents any sort of contamination between tests. So how does the MocheSeek technology work? First, we form a packed bed within the device. This packed bed consists of beads that are coated with our antibody of interest. Then we flow our chromatin through. The chromatin that we want will bind to the beads, and hopefully most of the ones we don't want will flow through. However, not all of the chromatin that we don't want will flow through. Some of it can bind, called non-specific binding. And so to remove that, we aggressively wash the beads using oscillatory washing. We use pressure alternating between the inlet and the outlet to aggressively wash the beads, as you can perhaps see in the video below. Finally, we use a magnet to pull the beads to one side so we can wash away any remaining non-specifically bound chromatin, which once that is done, we can elute the beads, remove the DNA, and perform sequencing. Well, how does MocheSeek compare to traditional ChIP-seq? First of all, we can obtain data with as few as 100 cells that is on par with data from 10 million cells. And each assay only takes 1.5 hours compared to overnight traditionally. And we can run 8 of them easily in parallel. So now that we have this technology that allows us to work with small amounts of cells and getting good data from it, we decided to look at the effect of BRCA1 in breast cancer. Now, 1 in 10 women will get breast cancer at some point in their life. Now, there's about 5 different kinds of breast cancer with various prognosis based on the kind. Of the women who are diagnosed before age 30, 25% of them have genetic causes. And of those 25%, 50% have a mutation in BRCA1. There's a 1 in 1,000 chance of having a BRCA1 mutation, but it confers about a 50 to 90% chance of getting breast cancer during their lifetime. They tend to have basal-like carcinoma, but they have a similar survival rate to those with basal-like carcinoma. However, they have higher rates of metastasis, recurrence, and mortality. So what does BRCA1 do that makes it so devastating if it's mutated? Well, BRCA1 is important for repairing DNA after double-stranded breaks. It's also critical in cell cycle checkpoint regulation. So for our study, we isolated breast tissue from women who were not carriers for BRCA1 or women who did have the BRCA1 mutation. Now, this is healthy breast tissue that we then separated into 4 cell types: mature luminals, luminal progenitors, basal cells, and stromal cells. Mature luminals and luminal progenitors are epithelial cells, and basal cells are myoepithelial, closely related, while stromal cells are fibroblasts and are the most different from the other 3. For each of these samples, we use our MocheSeek technology to profile them for H3K27AC, which is an enhancing modification, and for H3K4ME3, which is a modification associated with increased transcription or increased gene expression, higher gene expression. And we found that the H3K4ME3 didn't show many differences either between the non-carrier and the mutant, definitely not between the non-carrier and the mutant, and only slightly between the different cells. While on the other hand, H3K27AC, we see larger differences between the cell types, aside from luminal progenitors and mature luminals. This makes sense as luminal progenitors become mature luminals. And we generally see decent correlation between non-carriers and mutants for each cell type, except in the case of basal cells. We can also kind of see this when we analyze the peaks. Now, the peaks also show us that we see a difference in magnitude between the non-carrier and the mutant populations. A difference in magnitude generally means that one population or the other has far more modifications or far fewer modifications. So we analyzed these regions to determine which are differential regions, which ones are significantly different between mutant and non-carriers. And we found that basal cells and stromal cells had the most differential regions by far. The luminal progenitors had some differential regions, while mature luminals had almost none. This was actually a surprise for us, as while BRCA1 mutations are associated with a basal-like cancer, this basal-like cancer is shown to originate in the luminal progenitor population. So we expected the greatest differences to be occurring in the LPs. So what we then did was we linked these differential peak regions to nearby genes to determine if any pathways stand out that were changed that could help explain some of this. And we find that, well, there's a lot of pathways that are significantly changed. Those that are changed in both basal and stromal cells have to do with progesterone metabolism, RNA polymerase transcription, and actually the unfolded protein response. All 3 of these are associated with BRCA1. We also have basal cells, the ones that are only associated with basal cells, and we see lots of pathways associated with cell motility and adhesion. We also see epithelial commitment, which I will touch on again later, but the cell motility and adhesion also shown to be affected by BRCA1. And finally, in the ontologies only shown in stromal cells, we have antigen processing, apoptosis, and DNA damage response. Once again, all shown to be associated with BRCA1. So next we looked at enhancers. Enhancers are hotbeds of gene regulation. Transcription factors can bind to enhancer regions and regulate gene expression of nearby genes. When we called the enhancers, we found that the mutant population had far more enhancers in almost every cell type, except in stromal cells, which was interesting, but could be perhaps because the stromal cells were a completely different type of cell. And we do see differences in where these enhancers are located between the non-carriers and the mutants for each cell type, with the biggest difference shown in the basal cells. So, as I said, enhancers have transcription factor binding sites. So we determined which transcription factors were binding to these regions. And we found that the lowest overlap or the highest differences occurred once again in basal cells, with the mutant population having far more transcription factors, and in stromal cells with the mutant population having far fewer. The transcription factors involved, we had several that were strongly associated with BRCA1 that were also differential in multiple cell types, such as PAX5, CHOP, EGR1, and P73. We also analyzed the transcription factors using a pathway analysis, and we found pathways such as gonadotropic releasing hormone receptor pathway, Wnt signaling pathway, the apoptosis pathway, and P53 pathways. Now, the latter three are all very much associated with BRCA1. The first one isn't overtly associated with BRCA1. However, this pathway is a frequent target for treatment of breast cancer. So, as I mentioned before, generally, BRCA1 mutations are caused by an issue in the luminal progenitors, but we didn't really see that. We mainly see our changes in the basal cells. So we wanted to see what's happening to our basal cells. We looked at the cell type specific transcription factors, and if you can see the bottom row, that's the non-carrier basal cells. There's only a few cell type specific transcription factors. But if you look at the mutant basal cells, they have far more transcription factors, and many of them are the luminal progenitor specific transcription factors. To add on to this, 95% of basal cell specific genes that had differential marking between basal, non-carrier, and mutant samples had lower H3K27AC signal in the mutant. This suggests that the mutants were losing basal identity cells, suggesting that perhaps the mutant basal cells are becoming more luminal progenitor-like. And with that, I would like to thank my lab group, in particular, my advisor, Dr. Chang Lu, and my associate, Yunbang Xie. He is the one responsible for performing the MOWChIP assays. I'd also like to acknowledge Dr. Rong Li and Xiaowen Zhang from George Washington University for providing the samples, and our various funding sources, all of which from the NIH, for allowing us to perform this research. Thank you very much, and I'd be happy to take any questions.
Great. All right, so thank you so much. That was awesome. So I think we have a couple questions from the chat, and then for those watching, feel free to add more questions. So the first one is from Van Razel. And so the question here is basically, What are the implications of imperfect bead removal from the MocheSeek device?
Oh, you mean at the end of the assay? We generally find that there's are you saying from the end of the assay or the DNA from the beads?
That I'm not sure. It's asked, the question is basically, what are the implications of imperfect bead removal?
Okay, I will try and answer both aspects.
Okay.
The first one is whenever we remove the beads, we use a magnet to help move the beads along into the tube. We find that very few, if any, beads remain in the device. So if there is any beads remaining in there, I believe it would be comparable to having some material left on the sidewall of a micro tube. There's essentially no beads remaining there. As far as imperfect removal of the DNA from the beads, that would suggest that we would have less, but it wouldn't be targeted. So it would just be an overall reduction. So we shouldn't see any biases there.
Okay, so yeah, so the clarification was at the end of the assay when everything's all done.
Oh, okay, yeah, we pretty much grab like 99% of the beads, if not more.
Okay, great. All right, so this next question is from Ambika. And so the question is, does the vigorous washing potentially remove some of the chromatin of interest?
Yes, potentially. However, we did find that our MocheSeek technology was able to obtain like 50 times more DNA from a set of cells, from the same number of cells, than competing technology. The amount was actually within like the theoretical limit. So even if we are losing some, it's still far better than traditional ChIP-seq.
Great. So the next question is, what is the prep needed to go from tissue sampling to microfluidic processing?
Okay, so if you want a more in-depth article, we recently published in Nature Protocols, and that can give you a lot of assistance. But other than that, when it comes to the, for the breast work, we had the tissue, well, our collaborators had the tissue, they performed flow cytometry to separate the 4 cell types. Each cell type was then cross-linked and sonicated to get the chromatin, and then the chromatin was aliquoted and used in the microfluidic device. These microfluidic devices were created ahead of time, and that process of creating microfluidic devices is perhaps a day tops from complete scratch if you're making a whole new master and everything.
Great. So another question is, why is it important to separate the different cell types of the breast?
Yes. So what we found was that while there are some differences that you can find between the non-carriers and the mutants, when we compared our data with mixed populations, you can't really differentiate between the non-carriers and the mutants easily. We only see these differences once you actually split them out into the individual cell types. And we may have actually lost out on some of the nuances if we had used only mixed data.
So another question I had was in terms of the selection of, so there's a number of different types of microfluidic techniques for microparticle handling, different types of like washing stages and different types of arrays. How did your group decide on this particular one, the selection of magnetic beads and this particular mechanism of using a quake valve basically to serve almost like a filter in a way?
I guess it helps that our department we're in the chemical engineering department. So we drew a lot of our expertise from there. As you can see, a packed bed is very common to be used in chemical engineering processes. As far as beyond that, it was pretty much just trying to adapt a chip process to a device. And they knew that they needed to, once they got it through the packed bed, they saw that there was a lot of background. And so there wasn't as good of enrichment as possible. So they added in the oscillatory washing to help remove some of that background. I don't know if I have more specifics other than that.
Great. So for researchers who are potentially thinking about getting into this type of research, what do you think would be interesting next steps in researching BRCA1's role in breast cancer?
In particular, I think I would be most interested in two different aspects. If time and resources were not an issue, the first would be if we could do almost a timed course, especially if we can have it lead up until whenever they get breast cancer, because it's pretty much nearly a guarantee for many of these women, unfortunately. And another thing I'd be interested in is the breast cancer doesn't happen in both breasts at once. So if possible, I would get samples from both breasts so we can look at epigenetic changes between breasts, especially over time.
So, and then one last question for I know some of my undergraduate students will be watching. Do you have any general advice for undergraduate students who are thinking about graduate school in this kind of field? Any advice there?
So I guess my advice there is a lot of my coworkers had very little background in biology, and we do a lot of biotech work. And so my first piece of advice would be that don't be afraid to push yourself into new fields. I didn't know anything about microfluidics before I came here, and definitely not anything about the epigenome. So don't be afraid to push yourself into new fields. And the most critical part is the relationship with your advisor. If you have a bad relationship with your advisor, it's going to make grad life hell. If it's a good relationship, you'll enjoy the experience no matter how stressful it is.
So I take it back. We have one question from our president, Heidi. And so when the president asks, we keep going. So this will be the real last question. So what would you like to do next after graduating? You have a degree in computer science, a PhD in chemical engineering, with a lot of background also in biology. So what next?
I'm really interested in going into either industry or possibly government research. I would like to be able to bring all of my background together, ideally with either more bioinformatics work, either on the more computational side or more on the research side. And yeah, I'd like to really stay in with biology, with computer science, and be able to bring those two together. Okay, yeah.
Great. All right. Well, thank you so much for giving a great seminar today. And so for what we'll be doing is we'll update the description of the chat to include the article that Lynette mentioned during her talk and some other general information. And thank you again, Lynette, for such a great talk. And next week, we're going to have Sonia LeBlanc. And so please tune in next week as well. And we'll see you then. Okay. Bye, everyone.